Welcome back to the Deep Dive. Today we're looking at something that's really shaking up ecological monitoring, environmental DNA.
eDNA.
It's everywhere now, isn't it? This idea you can find out what's living where without actually seeing it.
Exactly. It pitched as this huge shortcut, finding out who's eating what, where things are, when they were there, all from traces they leave behind.
Like ecological forensics.
Kind of. And today we're doing a deep dive into just how reliable those traces are, specifically focusing on a paper led by Friedrich and colleagues.
Ah, yes. The one titled, "The quantity of deposited environmental DNA in plant arthropod interactions depends on taxon, abundance, and interaction time."
That's the one. And their mission was pretty critical. We know eDNA is great for just detecting if something is there, presence, absence.
Yeah, it's powerful for that.
But can we trust the amount of DNA, this read count they get back from sequencing? Does more DNA actually mean, more interaction, more bugs? Or is that signal just noise or biased somehow? And that's the absolute core question, the knowledge gap this study tackles head on. Using eDNA from plants, from leaves, seems so straightforward for tracking insects and what they're eating.
Just wash the leaf.
Pretty much. See who left their DNA calling card. But, and this is the big but, we haven't really understood how all the different factors, whether the insect itself, affect how much DNA actually gets deposited and sticks around.
So, without understanding those biases.
Exactly. Any attempt to compare populations or say this pest is more abundant than that one based just on DNA counts, it could be completely off. Meaningless, potentially.
Okay, so Friedrich and her team set out to pin down some of those potential biases. They took it out of the messy real world and into a controlled lab setting.
Which was essential.
And they focused on 4 key hypotheses, 4 factors they thought might skew the results. First, taxonomy.
Right, the type of insect.
Yeah. Does, say, a soft little caterpillar leave the same DNA footprint as a hard-shelled beetle? You'd think maybe not, right?
Intuitively, no. Their bodies, their behavior, it's all different.
Second factor, abundance. This seems obvious, but they had to test it. More bugs interacting with a plant. Does that actually lead to proportionally more DNA?
You'd hope so if it's going to be quantitative.
Third, interaction time. Does a longer visit, like a 6-hour munch fest, leave a stronger DNA signal than just a quick 1-hour stop?
Again, makes logical sense, but needs proving.
And finally, number 4, dietary preference. If an insect really likes a particular plant, its preferred food, does it leave more DNA there? Maybe because it's actually feeding, interacting more intimately compared to just like walking across a leaf it doesn't want to eat.
These are exactly the things ecologists grapple with in the field. If you're using eDNA counts to monitor, say, an invasive stink bug versus some native caterpillar, and you don't know if the stink bug naturally sheds 5 times more DNA just because it's a stink bug, your population estimates are fundamentally flawed from the start.
Right. You might think you have a massive stink bug invasion when maybe you just have.
Precisely. And field studies are just too chaotic to isolate these effects. Rain, wind, sun, degrading the DNA, you can't control for it all. That's why this controlled greenhouse experiment was so vital. It lets them really zero in on those 4 specific variables.
Okay, let's talk about that setup. It sounds like they were incredibly careful. Greenhouse experiments, Bayer AG in Germany.
Meticulous, yes.
They took leaves, washed them with deionized water, first, presumably to get rid of any existing DNA.
Exactly. You need a clean slate. A zero baseline. Otherwise, you're just measuring background noise, not the DNA deposited during the experiment.
Makes sense. Then these clean leaves went into sterile petri dishes with the insects, so only the DNA shed during that specific interaction time was measured.
That level of control is crucial for this kind of question.
Now, who were the stars of the show? The insects. They chose 3 quite different ones.
Good range, yeah.
First, the adult southern green stink bug, Nazara virigula. That's a hemipteran, piercing, sucking mouthparts, known for being a bit messy.
Definitely leaves a mark.
Second, the adult mustard beetle, Phaedon cochlearia. A coleoptera, hard body, different feeding style, chewing.
Represents a whole different order.
And third, the fall armyworm larva, Spodoptera frugiperta. L3 stage, so a soft-bodied caterpillar, a lepidopteran, completely different again.
So, Hemiptera, Coleoptera, Lepidoptera, adults, and a larva. Covers a good bit of ground taxonomically and developmentally.
And they manipulated those factors we mentioned. For the plants, they used Savoy cabbage, which they expected all the insects to like.
The preferred host, yeah.
Versus tomato, which was the less preferred host. The idea was they'd probably interact with both, but prefer the cabbage.
A good test of that dietary preference hypothesis.
And for abundance, they had a few versus many condition. But they adjusted the numbers for the insect size, which is smart.
Right, you can't just put 10 stink bugs in there. They're huge compared to the beetles.
So for the stink bugs, few was just 1 bug, many was 3.
Okay.
For the smaller beetles and armyworms, few was 3 individuals, and many was 9.
Makes sense. Keeping the biomass or interaction potential somewhat comparable.
And interaction time was that straightforward split.
Yeah.
1 hour versus 6 hours.
Simple comparison.
So the experiment runs, time's up. How do they collect the DNA?
They took the leaves, washed the surfaces again, but this time collected the wash water.
Okay.
Then they filtered that water through a very fine filter, 0.45 micrometers, to catch all the suspended cells, DNA fragments, whatever the insects left behind.
Right.
DNA was extracted from that filter paper. And then they used PCR to amplify a specific target gene, a standard marker, the mitochondrial COI gene fragment, about 210 base pairs long.
The barcode gene.
The barcode gene, exactly. They sequenced those amplified fragments, and the number of sequence reads they got back for each sample, that's their quantitative measure. The read count directly reflects how much insect DNA was recovered from the leaf surface.
Got it. So the moment of truth. The results. What did they find?
Well, first up, the good news, perhaps. All 4 factors, they tested the insect species, the plant type, the time, the abundance, all showed a statistically significant association with the amount of DNA recovered.
So the eDNA signal is responding to these things. It's not random.
Correct. It's detecting real biological signals. P-value less than 0.001. Very significant.
Let's break that down. What
Yeah.
worked as you might predict? Time and abundance.
Yes, those showed clear quantitative relationships. For exposure time, 6 hours resulted in significantly more DNA reads, an average of about 1154 reads,
Okay.
compared to only about 545 reads for the 1-hour exposure. So more than double the signal for 6 times the duration.
That's promising. And abundance.
Same story there. The many individuals condition yielded way more DNA, averaging almost 1,276 reads.
Wow.
Compared to just 412 reads for the few individuals conditions. So
Right.
roughly 3 times the individuals led to roughly 3 times the DNA.
Okay, that's actually really encouraging for using eDNA quantitatively, isn't it?
Yeah.
It suggests that, at least within a species, more time or more individuals does leave a stronger, measurable signal.
It absolutely does. It confirms that the potential is there. If you can hold other factors constant, the signal tracks these variables reliable.
What about the dietary preference, the cabbage versus tomato?
That also played out as hypothesized. They found significantly more DNA sequences on the preferred host, the cabbage averaging over 1080 reads,
Right.
compared to the tomato leaves, which only averaged about 660 reads.
And didn't they note something about feeding signs?
Yes, and this is a key detail. For the beetle and the armyworm, they observed feeding damage on the cabbage leaves, but none on the tomato leaves.
Ah, so it wasn't just preference, it was actual feeding behavior driving the difference.
It strongly suggests that, yes, the intimate interaction of chewing, maybe saliva, maybe frass production during feeding, leads to much higher eDNA deposition than just, say, walking across the surface.
Which makes eDNA potentially really good for confirming actual trophic links, who is genuinely eating what, not just who visited.
Precisely. That's a really valuable application, detecting those active feeding interactions.
Okay, so far so good. Time works, abundance works, preference works. But you mentioned a fly in the ointment.
Ah, yes. The big one. Despite those positive quantitative signals, the single most dominant factor influencing the DNA read counts was taxonomy, the species itself.
New type of insect.
By a huge margin. This taxonomic effect, this inherent difference in shedding rate between species, it essentially overwhelmed the other signals in terms of the magnitude of difference.
So wait, we expected maybe some difference between a beetle and a caterpillar, but you're saying the difference was massive.
Massive. Shockingly so, I'd say. The average read counts were dramatically different across the 3 species tested.
Okay, give us the numbers.
Right. The southern green stink bug, the Hemiptera, was the super shedder. It deposited by far the most DNA, averaging over 14-15 reads per sample.
Wow.
The mustard beetle, the Coleoptera, was in the middle, averaging about 835 reads. Still a fair amount.
Mm-hmm.
But the fall armyworm larva, the Lepidoptera, deposited way less, only averaging about 263 reads.
Hold on, let me get that straight. The stink bug left What, more than 5 times the amount of DNA as the armyworm larva?
Yes, over 5 times the signal from the stink bug compared to the larva, on average under the same conditions.
But the larva is soft-bodied. It's chewing.
Yeah.
You'd almost intuitively think it would leave more debris, more cells behind than a hard beetle or a sucking bug.
That's what makes it so counterintuitive, right? And the researchers were careful here. They checked for technical reasons, like maybe the PCR primers worked better for the stink bug DNA or differences in GC content affecting amplification. They ruled those out. The differences seem to be genuinely biological. It has to come down to the insects themselves.
Like what specifically?
Well, they suggest things like the amount or composition of saliva, maybe how much they move around on the leaf, and crucially, the physical properties and amount of waste they produce, their frass or fecal matter.
So the stink bug might just poop more or have stickier poop or something.
Something like that. Or perhaps its piercing, sucking, feeding style involving saliva injection and regurgitation is just inherently messier at a molecular level than the armyworm's chewing, even if the chewing looks more destructive visually. The adult stink bug simply leaves a much stronger, more detectable molecular trail.
Okay. This changes the picture quite a bit. What does this huge taxonomic bias mean for using eDNA in the real world?
It paints a picture of both great utility and significant limitation.
Let's start with the utility, the good news.
The good news is, if you're focusing on a single species, this study confirms plant eDNA is a really powerful tool. You can use the read counts to infer interaction intensity, more DNA. Likely means more bugs or longer interaction time for that specific species.
So if I'm monitoring the spread of just the southern green stink bug, for example, higher read counts probably do mean a bigger problem.
Exactly. It's great for species-specific monitoring and potentially for detecting those actual feeding interactions we talked about.
But the limitation?
The limitation is stark. This pronounced taxonomic bias means using eDNA for comparing across different species, like in a whole community survey, is incredibly problematic right now.
Because the loudest voice isn't necessarily the most abundant one.
Precisely. If you sample a leaf in the wild and get a ton of stink bug DNA reads and very few armyworm reads, you absolutely cannot conclude that stink bugs are more abundant or interacting more intensely overall.
It might just be that the stink bugs are champion shedders, and the armyworms are comparatively neat.
Exactly. It's like your earlier analogy trying to count a city population by footprints. If some people wear muddy boots and others wear clean slippers,
You're going to vastly overestimate the muddy boot wearers.
you are. Your sample is completely biased towards the loudest signal, the most efficient shedders, not necessarily the most numerous or ecologically important organisms.
So to make eDNA work for comparing across species in a community, we'd need, what? Correction factors.
That's essentially it. We'd need species-specific shedding coefficients. We'd need to know, roughly, how much DNA does an average stink bug shed per hour compared to an average armyworm, compared to an average beetle, and so on, for potentially hundreds or thousands of species.
Wow. That sounds like a monumental task.
It is, and this study provides the first quantitative glimpse into just how different those shedding rates can be. That 5-fold difference is substantial. Future work needs to expand this kind of controlled experiment across many more taxa.
Are there other factors they mentioned needing investigation?
Yes, they flag things they couldn't test. Abiotic factors, for one. How do temperature and rainfall affect shedding and DNA persistence on the leaf? Does heat make insects shed more? Does rain wash the signal away or maybe even help spread it?
Good points.
And also more biological detail about the arthropod itself. Its life stage, does a pupa shed differently than an adult? Does a highly mobile species leave more trace than a sedentary one? All these could further modulate the signal.
So wrapping this up, the big aha moment from this paper is what?
I think it's this. eDNA from plants does carry a clear quantitative signal. It reflects interaction time, it reflects abundance, which is fantastic.
Right.
But that signal is filtered or distorted very strongly and very differently depending on which species left it. It's a powerful lens for looking at one species at a time, but it's like a funhouse mirror when you try to look at the whole community simultaneously without calibration.
It tells you how much is happening for a known bug, but it doesn't give you a balanced picture of all the bugs without a huge amount of extra work.
Exactly. And think about what that calibration truly entails, given that 5-fold difference they found just between two common insects. Imagine the complexity. To really use eDNA for unbiased, community-wide monitoring across diverse groups, you might need correction factors not just for the species, but maybe for temperature, for life stage, for whether it was raining. It gives you a framework for just how sophisticated and complex this powerful tool really is. We know it works, but making it work accurately across the board, that's the next major frontier.